Hello everybody. Today I will be talking about my bioinformatics project, genetic identification and characterization of unknown Rouxiella bacterium. So this project started from a bacteria isolated in Bio 378 microbial diversity by a student, Gianna Di Francesco. This bacteria was isolated from a decomposing strawberry and cultured. So this bacteria falls under the Enterobacteriaceae family, which is a family of gram-negative bacteria. This family is very familiar to humans for its relevance in diseases, as many bacterial pathogens fall under this family. So some basic facts about it: it is a rod-shaped bacterium, as you can see from this image right here. And really, that's about all that's known of it. Very little is known about this genus, as it was discovered back in 2013. The first species to be identified was Rouxiella chamberdiensis, and it was isolated from parenteral nutrition bags, which are feeding bags that you have when you're in the hospital. It's tube feeding, essentially. And so this bacteria was implicated in the deaths of 3 infants in France. The other 2 bacterial species of this genus were discovered in Germany, one in a peat bog and one in a meadow. So right off the bat there, this is a very cosmopolitan bacteria. It's found on a strawberry in a hospital setting and also just in the wild, which makes it quite interesting. Gianna had isolated this and gone through the process of sequencing its genome, but other than that, nothing was known about the bacteria. So the purpose of all of this is to identify what it is, whether or not it's new, and all of that. So the methods that we followed a pretty traditional bioinformatics pipeline initially, and then as the project became more specific and it became more apparent that what we're working with here was novel, we took 6 specific tools. We used a program called QUAST, which is a quality assessment tool for genomic assemblies. A really great tool for visualizing your assembled genome. Then we used something called PROCA, which is a whole genome annotation piece of software done in a command line environment. It's very useful identifying genes and specific features of a genome, and is an automated piece of software. Then we use something called RAST, which is a piece of software that is online, and that is the Rapid Annotation via Subsystem Technology. And this is a piece of software which is an automated pipeline that annotates the entire bacterial genome and provides metrics and information about the genome. So using the information gathered from those 3 programs, we used 2 different types of BLAST searches. We used a local BLAST done in the command line environment, as well as the NCBI BLAST, which utilizes the NCBI's BLAST database. And then finally, all of the information that we gathered related to specific genes was put to a program called MEGA, which is the Molecular Evolutionary Genetics Analysis. And what it did for us was to provide some very nice phylogenetic trees. So with the results, starting here with table 1, this is the GC content, or the guanine cytosine content of the bacteria. It's the percentage of nitrogenous bases on a DNA or RNA molecule, and that are either guanine or cytosine. And GC content is species specific. So it's almost like a barcode. If you have a GC content percentage, you can match that up to its correlating bacteria. So as we can see here, our Rouxiella had a GC content of 52.74%, which is different than the other 3 known Rouxiellas. So that was initially very exciting because that was the first hint that this was in fact a new species. And then we just have some other general metrics about the bacteria. This graph right over here shows the cumulative genome length as generated by QUAST here. This is one of the graphs that program gave us. and it is comparing the length of this genome from our isolate to the known genome of Rouxiella chamberdiensis, so a known species. Basically showing that they're more or less the same length, which is a decent indicator that it's at least in the same genus of bacteria. So this pie chart right here is just showing the functional composition of the coding sequences found in the DNA. So essentially, this percentage of the genome right here correlates to a specific function, be it carbohydrate synthesis or other RNA syntheses and biological processes, making proteins essentially. And then this table right here is just a general overview of our organism, our Rouxiella species, as generated by RAST, more or less just repeating what the other three images show. So the big part of our results is this image right here. This phylogenetic tree was created using the RPOB gene. So the way these trees were created was we found the sequence for a specific gene in our Rouxiella and performed a blast search on it to find similar sequences in different organisms. And from there, we gathered all of those different sequences, aligned them, and using Mega, it produced this tree. So as you can see right here, our Rouxiella is different based off the RPOB gene in comparison to other bacteriums inside the Enterobacteria family. We did this for two other genes, the SUC-A and the FUSE-A gene, which those results are not shown here. However, they gave a very similar phylogenetic tree to this one here. So moving to this phylogenetic tree, this one was done using sequences from the 16S gene. Now, traditionally, the 16S gene is very great at being a species identifier for bacteria. Every bacteria has a 16S gene, and so usually you can determine what it is by looking at the 16S. However, it's not good for comparing bacteria from the same family because the sequences are so similar. It's not statistically relevant enough to determine its identity. And so essentially that is all this graph here is showing, is that we were able to construct a tree using the 16S sequences from all these bacteria, but the length of the branches and all the mathematics corresponding with it are not significant enough. So essentially, overall, based off of the GC content and our phylogenetic trees, the data is sufficient enough to say that what we have here is a new species of bacteria, which is very quite exciting. So essentially future directions for this before this species of bacteria can be officially designated and amended into the list of known bacteria. Much more work has to be done. There has to be a lot of wet lab and phenotypic testing, but this is the first major step in saying that we have a new species of bacteria.